Welcome to Rad Onc Smart Review Rad Bio Edition.
Hey everyone, we're excited to spend some time with you today, really getting into a topic that's, well, it's rapidly changing how we think about cancer therapy.
That's right, we're talking about the intricate relationship between radiation and the immune system.
Absolutely. For a long time, radiation was seen primarily as this local weapon, just directly kill cancer cells.
Yeah, just zap the tumor. But we now understand its role is much broader, its powerful role in modulating the immune system and the tumor microenvironment.
Understanding this synergy is so key, especially now as we combine radiation with, exciting new therapies like immune checkpoint inhibitors. It's not just about targeting the tumor directly with radiation anymore. It's also about sort of teaching your body to fight the cancer too.
And this is really high-yield material for your board prep. So we're here to walk you through the essential concepts, the clinical relevance, and, some crucial points to remember.
Think of this as a focused tutoring session. We want to help you feel confident with this stuff.
Okay, so let's unpack this. We should probably start with the absolute basics of immunity.
Good idea. Our bodies have essentially two main defense systems working together: innate and adaptive immunity.
Right. Innate immunity, that's your body's first responder, isn't it?
Exactly. It's nonspecific. It kicks in really quickly when it detects something foreign or damaged.
Like physical barriers, skin, obviously,
Okay.
and then those phagocytic cells, macrophages, neutrophils.
Yep. And natural killer cells, or NK cells, the complement system. They recognize these broad patterns on pathogens, or what we call damage-associated molecular patterns, DAMPs. Those come from injured cells.
DAMPS, got it. Okay, then you have adaptive immunity.
Right. Now, this system is much more sophisticated. It's highly specific.
Right.
And crucially, it remembers past encounters.
Takes a little longer to get going, though.
It does, yeah. But it provides that long-lasting, targeted protection.
And the main players here are lymphocytes, B cells and T cells.
Right. B cells handle humoral immunity. They make antibodies, those proteins that tag specific invaders.
And T cells, they're all about cell-mediated immunity. You've got the C8 positive T cells.
The cytotoxic T lymphocytes, CTLs, they're like the assassins, They directly kill infected cells or cancer cells.
And the CD4 positive T cells, helper T cells.
Yeah, they're like the conductors of the orchestra. They coordinate the whole immune response, including helping activate those CD8 positive T cells and the B cells.
And we shouldn't forget regulatory T cells, Tregs.
Definitely not. Tregs are crucial for preventing autoimmunity, basically for keeping the immune system from attacking healthy tissue by suppressing responses.
Okay, so for those T cells, especially the CD8 positive killers, to actually recognize a cancer cell, they need to be shown what to look for.
Precisely. And this is where antigen presentation comes in.
Antigen-presenting cells, APCs, they're key here.
Right. Dendritic cells, or DCs, are the most effective APCs, the real professionals. But macrophages and B cells can pitch in too.
So the process starts with antigen uptake and processing.
Yep. The APC basically engulfs something, maybe a pathogen or maybe a dying cancer cell that contains tumor antigens. Then it breaks it down inside into small peptide fragments.
And then the crucial step, MHC loading and presentation.
Exactly. And there are two main types of these MHC molecules we need to know.
Okay. First, MHC class 1 molecules. They're found on pretty much all nucleated cells, right?
Almost everything, yeah. And they display peptides that come from inside the cell. Things like viral proteins if a cell's infected, or, importantly for us, those mutated cancer proteins.
And CD8 positive T cells recognize these.
That's their job. So if a CD8 positive T cell sees a tumor antigen peptide sitting on MHC class 1,
That's the signal. Tells it that cell needs to be destroyed.
bingo, marks it for destruction.
Okay, so MHC class 1 shows internal stuff to CD8 T cells. What about MHC class 2?
MHC class 2 molecules, they're primarily on those professional APCs like the dendritic cells.
Right.
And they display peptides from stuff the APC engulfed from outside the cell.
Okay, external antigens.
Exactly. And these are recognized by the CD4 positive helper T cells.
And activating those helper T cells is vital for a strong overall immune response, right? Because they help everyone else.
Couldn't have said it better. They're essential coordinators.
Okay, so the T cell finds its antigen on the MHC molecule. But that binding, signal 1, isn't enough for full activation.
Correct. For a T cell to really get fired up, it needs a second signal from the APC. Signal 1 is that specific binding the T cell receptor, TCR, engaging with the peptide MHC complex.
And signal 2 is co-stimulatory.
Right. It involves other molecules. On the APC, you have things like the B7 proteins, also called CD80 or CD86. These bind to receptors on the T cell, like CD28.
It's like a confirmation signal. Yes, this is real danger.
That's a great way to put it. It confirms there's a real threat, and the T cell needs to mount a full attack. Without that second signal, the T cell might become energetic, basically unresponsive, or it could even die. It prevents accidental activation.
Makes sense. Okay, so the immune system is incredibly powerful, but it needs checks and balances.
Absolutely. You need ways to stop it from attacking your own healthy tissues. And these are the immune checkpoints. They're essentially brakes on the system.
And the problem is, cancer cells have figured out how to exploit these breaks to hide.
Unfortunately, yes. They've learned to manipulate these checkpoints to escape detection and destruction.
So two of the most important ones we need to know, especially for the boards, CTLA-4 and the PD-L1 pathway.
Exactly. Those are critical. CTLA-4 is primarily involved earlier on in the initial phase of T-cell activation, often happens in the lymph nodes.
Okay, early activation phase, lymph nodes, and it's expressed on activated T cells.
That's right. And the key thing about CTLA-4 is that it binds to those B7 molecules on the APCs more strongly than the activating CD28 receptor does.
Ah, so it outcompetes CD28.
Precisely. So when CTLA-4 is present and binds B7, it wins that competition. And instead of an activation signal, it delivers an inhibitory signal. It dampens down T-cell activation and proliferation.
So blocking CTLA-4 with antibodies, like ipilimumab, ipilimumab,
Right.
that basically releases this early break.
You got it. It allows T cells to get activated more strongly right from the start.
Okay, that's CTLA-4. Then there's the PD-L1 pathway. You said this is more critical in the tumor microenvironment.
Yeah, this interaction often happens right where the T cells meet the tumor cells. PD-L1 is an inhibitory receptor found on activated T cells, but also on B cells and NK cells.
And its main partner, its ligand, is PD-L1.
Correct, and PD-L1 can be expressed by the tumor cells themselves or by immune cells within the tumor microenvironment.
And often that PD-L1 expression goes up in response to inflammation, like interferon gamma from T cells, right? That adaptive resistance thing.
Exactly. The T cells try to attack, release interferon gamma, and the tumor responds by putting up more PD-L1 shields.
So when PD-1 on the T cell binds to PD-L1 on the tumor cell,
It's an off switch for the T cell. It tells the T cell to stand down. This leads to T cell exhaustion or anergy, basically lets the tumor evade the attack.
right, so antibodies blocking PD-1 like nivolumab, or pembrolizumab,
Or blocking PD-L1 like atezolizumab, durvalumab, or ovolumab.
they disrupt that stand-down signal.
Precisely. They essentially reinvigorate those exhausted T cells, take the brakes off right there in the tumor so they can get back to fighting.
Okay, great overview of the checkpoints. Now let's tie this back to radiation oncology. We know radiation kills cells, but how does it interact with this whole immune picture?
Right, because it's far more than just local cytotoxicity. Radiation profoundly affects the tumor microenvironment and interacts with the immune system in really complex, interesting ways.
This is where it gets really interesting, I think. Radiation can actually boost anti-tumor immunity.
It really can, through several key mechanisms. First and foremost, it causes tumor cell death, and when tumor cells die, they release stuff.
Like tumor-associated antigens, TAAs, and maybe even new ones, neoantigens from radiation damage.
Exactly. But crucially, radiation can induce a specific type of cell death called immunogenic cell death, or ICD.
ICD. Why is that special?
Because ICD involves the release or exposure of those damage-associated molecular patterns, those DAMPs we mentioned way back at the beginning, danger signals.
Ah, okay. Like what kind of DAMPs?
Key ones include calreticulin, CRT, that moves to the cell surface and acts like an eat me signal for dendritic cells.
Okay.
Then there's ATP, which gets released and acts as a find me signal, attracting APCs to the site.
Find me. Got it.
And HMGB1. It's released from the nucleus and binds to toll-like receptors, TLRs, on APCs, helping them mature and present antigens more effectively.
So these DAMPs aren't just debris, they're active signals.
Absolutely critical signals. They promote the uptake of tumor antigens by APCs, drive their maturation, and encourage them to migrate to the lymph nodes to prime T cells. Turns the dying cell into an immune stimulant.
Okay, that's a huge one. What else does radiation do immunologically?
Second, radiation can actually increase the expression of MHC class I molecules on surviving tumor cells.
Oh, right. We said MHC1 shows internal antigens to CD8 T cells, so more MHC1 means.
Means those surviving tumor cells are more visible targets for any CDD8 positive cytotoxic T cells that have been newly primed against tumor antigens. Makes them easier to spot and kill.
Nice. And third,
Third, radiation can modulate the tumor microenvironment, the PME. It can alter the mix of signaling molecules, the cytokines. Sometimes it can shift a suppressive environment to be more pro-inflammatory.
allowing immune cells to get in easier.
Exactly. It can help promote the infiltration of those beneficial CD8 positive T cells right into the tumor.
But, and this is important, you mentioned it's a double-edged sword.
It absolutely is. Radiation isn't always pro-immune. It can also have immunosuppressive effects.
Like what?
Well, it can sometimes increase the recruitment of those suppressive cells, like Tregs or myeloid-derived suppressor cells, MDSCs. Or, somewhat ironically, it can even upregulate PD-L1 on tumor or immune cells.
The very thing we try to block with checkpoint inhibitors.
Exactly, so it's complex.
But that complexity kind of highlights why combination therapy is so promising, doesn't it?
It really does. By killing tumor cells and altering the TME, maybe radiation helps reduce that chronic antigen exposure that contributes to T-cell exhaustion.
Setting the stage for the checkpoint inhibitors.
Precisely. Radiation can prime an anti-tumor response, releasing antigens, DAMPs, upping MHC1, changing the TME.
And then the checkpoint inhibitors, the anti-CTLA4, anti-PD-1, anti-PD-L1, they come in and release the brakes on that now primed immune response.
And the result, hopefully, is a much stronger, more durable anti-tumor effect than either treatment could achieve on its own. That's the basis for so many clinical trials right now.
Okay, let's touch on two other phenomena that sometimes get confused. The abscopal effect and the bystander effect.
Good call. They're related to radiation's broader impacts, but are distinct. The abscoichal effect, that's the regression of metastatic tumors at sites distant from the primary tumor that was actually irradiated.
So you treat one spot and another spot shrinks even though it didn't get radiation.
Exactly.
Huh?
And it's thought to be purely immune-mediated.
How?
Well, the idea is that radiation to that primary site does everything we just talked about, releases antigens, DMPs, activates APCs, primes T cells in the lymph nodes.
And those activated T cells then circulate systemically.
Right. They travel through the bloodstream, find tumor cells at those non-irradiated metastatic sites, recognize them, and attack.
Historically, this was super rare, right? Like almost a medical anecdote.
Very rare, yeah. A curiosity. But now, with effective immunotherapies, especially checkpoint inhibitors,
We're seeing it more, or at least there's more interest.
renewed interest, definitely. And maybe slightly more frequent reports. It seems that combining radiation with checkpoint inhibitors significantly increases the likelihood of seeing an abscopal response.
Okay, so abscopal is systemic, immune-mediated, affects distant sites. How is the bystander effect different?
The radiation-induced bystander effect is different. This refers to biological effects, things like DNA damage, changes in gene expression, maybe even cell death happening in cells that were not directly hit by the radiation beam.
But are nearby the irradiated cells.
Exactly. Or they receive signals from those irradiated cells.
What kind of signals?
It's mediated by stuff released from the irradiated cells. Reactive oxygen and nitrogen species, ROS and RNS cytokines like TGF-beta, and TNF-alpha. Tiny packages called extracellular vesicles like exosomes, even direct communication through gap junctions between adjacent cells.
So it's more of a local neighborhood effect.
Generally, yes. The significance is that radiation's biological impact isn't just strictly within the dose lines we draw. It can extend slightly beyond, potentially contributing to both therapeutic kill of the tumor edge and maybe some normal tissue toxicity too.
Its exact clinical roles is still being figured out.
Still under active investigation, yeah.
Okay, perfect distinction. Let's shift now to some clinical pearls. These are the absolute high-yield nuggets you need to nail for your board exams.
All right, let's crystallize the key takeaways.
Pearl number 1, remember the fundamental difference. Innate immunity. Fast, non-specific. Adaptive immunity. Slower, but specific and provides memory.
Got it. Pearl number 2. Antigen-presenting cells, especially dendritic cells, are absolutely key. They present antigens on MHC1 to CD, 8 positive T cells, and on MHC2 to CD, 4 positive T cells. Know which is which.
Pearl number 3, know your immune checkpoints. CTLA-4 acts early during T-cell priming, often in lymph nodes, competes with CD28 for B7. PD-L1 acts later in the tumor microenvironment, leads to T-cell exhaustion. Blocking them releases the brakes.
Essential. Pearl number 4. Understand how radiation boosts anti-tumor immunity. Key mechanisms: releasing tumor antigens via immunogenic cell death, releasing DAMPs like calreticulin, that's your eat meat signal, HMGB1, and ATP+, upregulating MHC1 on tumor cells and modulating the TME.
Pearl number 5. Clearly differentiate the abscopal effect, that regression of distant, non-irradiated tumors, immune-mediated, more likely with immunotherapy, from the bystander effect, which involves effects in non-irradiated cells near irradiated cells due to release signals, systemic versus local.
Perfect. And pearl number 6, be familiar with the common checkpoint inhibitors and their targets. Ipilimumab targets anti-CTLA-4, nivolumab and pembrolizumab target anti-PD-1, and atezolizumab, durvalumab, and avelumab target anti-PD-L1. Know that list.
Excellent pearls. So, translating this, what does it all mean for clinical practice today and going forward?
Well, the understanding that radiation can act like an in situ vaccine, priming an immune response right there within the patient, that has really revolutionized how we think about combination therapies.
It provides a really strong rationale for combining radiation, especially techniques like SBRT, stereotactic body radiation therapy, which gives high doses, good for cell kill and antigen release.
Combining that with immune checkpoint inhibitors like the anti-PD-1s or anti-CTLA-4 drugs.
And the hope is to take immunologically cold tumors, the ones without many immune cells,
and turn them hot. Boost T-cell infiltration, prime systemic immunity, maybe even trigger those abscopal effects for better control of metastatic disease.
But as you said, this field is still evolving really fast.
Incredibly fast. There are still significant questions we need answers to.
Like optimal timing.
Yeah.
Dose. Fractionation of the radiation when you combine it. Sequencing.
Exactly. Concurrent versus sequential. What's the best radiation schedule for immune priming versus maybe causing immunosuppression? How do we manage the combined toxicities? It's not straightforward.
And not all tumors or patients respond equally well.
Right, which highlights that urgent need for biomarkers. We need ways to predict who's most likely to benefit from these powerful combinations and how to personalize the approach.
Okay, ready for a quick board blitz? Let's see if we can apply some of this.
Bring it on. Let's test that recall.
Question number 1. Which molecule, when exposed on the surface of irradiated tumor cells, acts as an eat-me signal, promoting phagocytosis by dendritic cells? Is it AHMGB1, BATP-CPDL1, or D-calreticulin?
The eat-me signal. That would be D-calreticulin. Its translocation to the cell surface is a key DAMP for signaling dendritic cells to engulf the dying tumor cell.
Correct. Question number 2. A patient with metastatic melanoma receives radiation therapy to a single liver metastasis. Subsequently, regression is observed in non-irradiated lung metastases. This phenomenon is best termed A. Bystander effect, B. Radioresistance, C. Abscopal effect, or D. Adaptive resistance.
Okay, radiation to one site, regression at a distant, non-irradiated site. That's the definition of C. Abscopal effect, mediated by a systemic immune response.
Spot on. Question number 3. The PD-1 receptor is primarily found on which cell type, and its interaction with PD-L1 typically leads to what outcome? A. Tumor cells, T-cell activation, B. Dendritic cells, T-cell priming, C. T-cells, T-cell anergy or exhaustion, or D. Regulatory T-cells, B-cell activation.
PD-1 is primarily on T-cells, activated T-cells, and its interaction with PD-L1 leads to inhibition. So C T-cells, T-cell anergy or exhaustion.
Exactly right. And finally, question number 4. Which of the following is not a primary mechanism by which radiation therapy can enhance anti-tumor immunity? A. Release of tumor-associated antigens from dying tumor cells. B. Induction of immunogenic cell death, or ICD, and DAMPs. C. Upregulation of MHC class I on tumor cells. Or D. Direct activation of B cells to produce tumor-specific antibodies.
Okay, which is not a primary mechanism. Radiation causes antigen release. A. Induces ICD and DAMPs. B. Upregulates MHC1AC. Direct, large-scale B cell activation. D isn't really considered a primary direct effect in the way the others are, so D.
Correct. While B cells might get involved downstream, it's not a primary radiation mechanism like the others. Well done.
Good questions. They really hit the key points.
So as we discussed, one of the biggest areas of active investigation, maybe even controversy right now, is figuring out the optimal way to combine radiation and immunotherapy.
Absolutely. It's not just about throwing them together. Is concurrent better? Sequential? What dose, what fractionation schedule for the radiation is ideal for immune priming? Maybe it's different from the dose for maximal local kill.
And how do we balance that against potential immunosuppression from radiation or the combined toxicities?
Huge questions. And the search for those predictive biomarkers is just critical. Imagine being able to know upfront if a patient's likely to have an abscopal response, or who will benefit most from adding immunotherapy to their radiation.
That's the goal. Personalized immunoradiotherapy.
That's where the field is headed.
Okay, let's try and bring it all together then. Quick summary.
That sounds good. Today, we've done a pretty comprehensive review of how radiation interacts with the immune system, a really dynamic area.
We covered the basics, innate and adaptive immunity, antigen presentation via MHC molecules, and the vital role of those immune checkpoints, CTLA-4 and PD-1, PD-L1, in regulating T-cell responses.
Then we really dug into how radiation therapy is so much more than just a cell killer. It can effectively transform the tumor into an in situ vaccine.
Right, by promoting antigen release, inducing immunogenic cell death, with those key DAMPs, calreticulin, HMGB1, ATP upregulating, MHC1, and modulating the tumor microenvironment.
And these immune-modulating effects provide that strong foundation for combining radiation with immune checkpoint inhibitors. The goal is synergistic anti-tumor activity.
Potentially leading to that fascinating, though still relatively rare, abscopal effect. And we made sure to distinguish that from the more local bystander effect.
Understanding these concepts, these pathways, it's just fundamental, not only for treating patients effectively in the future, but really for mastering your board exams.
Keep reviewing these pathways and effects. You know, you're building a really solid foundation, and every study session, honestly, brings you closer to your goal. We know this material is complex, but breaking it down like this, step by step, makes it manageable. You've absolutely got this.
Totally agree. And looking ahead, in our next episode, we'll start a high-yield review of key signaling pathways relevant to radiation biology.
You can complete practice oral boards at radoncsmartlearn.com and subscribe to Rad Onc Smart Review for our next episode.
Thanks, everyone, for tuning in.